Hello friends, myself Nilofer Abid Khan, Assistant Professor from Kukanyanpet Rahul Dakar College of Pharmacy and Research Institute, Karza. Today we are going to see steroids, cardiac glycosides and triterpenoids introduction. Let's begin. Triterpenoids are the compound with carbon skeleton and possess 6 isoprenyl unit. They are biogenetically derived from squalene, that is acyclic 630 hydrocarbons. They are generally a complex cyclic structures of alcohol, aldehydes or carboxylic acids. They are generally have a high melting point and they are optically active. Colorless in nature with crystalline structure. To recognize or to characterize these compounds are quite hard because they possess less chemical reactivity. These triterpenoids are divided into four broad groups such as true triterpenoids, steroid, saponin and cardiac glycosides. Now let's see what is sterol. Sterols and triterpenoids, they contain cyclopentane per hydrophenanthine ring. You can see this structure. This is phenanthine ring which will be having an hydrogen, so it is less, double bond is not there, only one single double bond is present at this end, and here you can see a cyclopentane ring. So it will be called as cyclopentane per hydrophenanthine ring. This is the structure of sterol. Sterols primarily are considered as an animal products such as sex hormones, bile acids, etc. But later their occurrence were also reported in plant tissues. There are three phytosterols, that is sitosterol, which is also known as beta sitosterol, sigmasterol, camphosterol, which are generally seen in higher plants abundantly. These sterols occur either in a free form or as a simple glycosides. Some examples of these sterols are alpha-spinasterol, which will be found in spinach, alfalfa and seneca. Ergosterol, which will be found in fungi and yeast. Flucosterol, which will be found in brown algae and coconut. Now let's see what is saponin. Saponins are the glycosides of triterpene and sterol. They occur in more than 70 families of plants. They have soap-like properties and act as in surface active agents. They have an ability to hydrolyze the blood cells. Some examples of saponins are hecogenin from agave, yamogenin from Dioscorea, saponins of alfalfa and glycyrrhizine of licorice. The glycosidal pattern of saponins are quite complex. They have as many as 5 sugar units attached and a glucuronic acids. These saponins will be divided into two categories. That is, means saponins is made up of two parts, that is glycon and aglycon part. In that, glycon part A will be either a glucose, it can be an arbinose, xylose or glucuronic acids. A glycon part will be sapogenin. Now let's see what is cardiac glycosides. Chemically, cardiac glycosides, a glycon part of cardiac glycoside is a steroidal moiety. This steroidal moiety may be either a C23 or C24 unit because of either 5-membered or 6-membered lacton ring respectively. The presence and attachment through a specific position to the steroidal nucleus of a lacton is essential for a proper cardiac activity. The sugar moiety will be attached through C3O linkage. Various types of sugars are reported in cardiac glycosides. They are as glucose, fructose, rhamnose, digitoxose, saccharose and so on. Though the sugar moiety does not have any medicinal activity, but still they will be useful for solubilization of glycon, for beneficial in absorption and distribution of glycon moiety. At the time, up to 3 sugar molecules can be attached to an glycon moiety. It has been also shown that increasing the number of OH group on a glycon leads to quicker onset of action and enhance the metabolism. Congestive cardiac failure, which is consequence of different diseases, treated by cardiac glycosides. They act as cardiotonic by increasing the systolic contraction satisfactory. Here you can see the structure of cardiolinoids and bufadienoids. Okay, so this is hydrophyllanthrene with a cyclopentanes, and here you can see a 5-membered lacton ring and a 6-membered lacton ring. This is cardiolinoids and this is bufadienoids. Now let's see what is the difference between cardiolinoids and bufadienoids. Cardiolinoids occur in a region is quite high as compared to that of bufadienoids. They have a 5-membered rings and they are also called as butyrolacton rings. The lacton rings of these cardiolinoids will contain only one single bond. You can see in this picture, in the structure, that it will be a one single double bond, and here the attachment of the steroidal nucleus through it attached the steroidal nucleus by C17 position. Which are the families which belongs to cardiolinoids like Leguminosae, Sterculariaceae, Cruciferaceae or Scorpulariaceae are the families which will be shown in the which shows cardiolinoids. Now let's see what is bufadienoids. Bufadienoids are the name term is derived from bufalene, that is obtained from the skin of toads. They contain 6-membered lacton rings. You may call also this ring as pyron ring. Here you can have 2 double bonds and they attach to the steroidal nucleus through 17 beta position. Which are the families? They are seen in very few families like Liliaceae and Rankulaceae. Thank you.